I would say that right now it's kind of too preliminary because the numbers are too small. But as we do more and more of this, I think we'll get a much better idea. We get samples from people in their 60s and 70s more rarely than we do relatively younger people. So it'll take more time. But we definitely see changes in gene expression for neuronal genes within the sample that we have right now, which I think is promising and also very interesting. So we're looking forward to doing more of that. The second question, we have not looked if there are differences in pain qualities between the men and women having thoracic vertebrectomy. But we should do that, actually. I don't know why we haven't thought of that before. In the C1, C2 DRG, C1, C2 fusion study that we're doing, we're doing a far more kind of aggressive QST profiling. And we can do that because these patients aren't nearly as complicated. They don't have relatively advanced cancer, et cetera. So I think there we might have a better opportunity as that project progresses to see how there might be sex differences in pain qualities that correlate to underpinning mechanistic differences that might be driving pain. So as that project really gets rolling, as we start to pull in more data, I think that would be a really great thing for us to look at.
Cool. Thank you.
Yep.
Thanks, Anne. Okay, I'm going to go to some of the questions in the chat, try and go in order. Very simple, straightforward question from Bob Coghill. Based on the different molecular signatures you see in the DRG, which is the better model for the human, rat or mouse? And then someone, I think, was tagging onto that question along those lines. Are there noticeable strain differences within mice or rats?
Oh, well, there's a huge literature on the strain differences. So there definitely are. In terms of which is a better model, I think, the thing that this data allows you to do is to make that determination depending on the target that you're looking at. So, if you're interested in, let's say, interferon signaling in a rodent model, which I actually am, you can choose models where the changes in interferon signaling correlate well with what we see in the human DRG samples from the thoracic vertebrectomy patients, for instance. And also, whether or not there's a sex difference. If you're interested in a particular target, that is expressed in nociceptors in humans, but it's not in mice, but it is in rats, obviously rat is the better choice, right? So I think we're at the point now where we have this data where you can make those kind of judgments relatively quickly. In terms of which species, better represents nociceptor subtypes kind of, holistically, I don't think that mice represent it very well. And rats, we don't yet have the kind of equivalent data that we have in mice to make that kind of judgment. But based on kind of the history of the literature, I would say rats are likely to have kind of better representation of human nociceptor subtypes.
Thanks, Ted. I'm going to keep going since we have some more questions. We want to try and get through some more. So from Kathleen Sluka, fascinating data. Could the differences you find with A2CPS and differences in transcriptional regulation explain the lack of translatability of some of the targeted drugs from rodents to humans, like TRPV1 antagonists or substance P NK1 antagonists? And then she has a separate question. The sex differences are incredibly important and completely understudied. Will you take that data from humans that are unique to male and female and test them in rodents, or do you believe it's too difficult to translate from rodent to human to do that work?
Right, I'll start with the sex differences question first. I mean, what we can do now, because we can do these human DRG dissociated cultures and explants, is we can use the signaling mediators that we see from the pain patients, and then put these on DRGs from organ donors and see if there's a sex difference in how they signal, etc. So Juliet, who's one of the PhD students in my lab, she's kind of actively working on a project along those lines, and I think that that's the direction that we'd like to go from that perspective. On the first question on the ATAC-seq, I think what that allows us to identify is, what is the underlying reason that there are these major species differences, it should be driven by some transcriptional regulation. And I think we're starting to uncover how that likely happens.
All right, thank you. Michael Hildebrand has a question. He did also say thanks for the shout out earlier.
Yeah,
excellent talk,
sure
Ted.
thing.
Really interesting finding that CGRP expression is elevated in itch DRG neurons in female compared to male humans. A quick question on how these DRGs are identified as itch specific in the human DRG samples based on markers previously identified from rodents, or?
Yeah, they are based on markers identified from rodents, and also receptors that are expressed in that population that we know induce itch in humans. So that's how we did that. And I do have to teach Stephanie.
Oh, I'm sorry. We just have one more hand up. But
yeah, I can take one more question.
Dr. Zhang Chang, if you want to quickly. Sorry.
Sure, very quickly. Very interesting observations. For the spinal cord neuronal clusters, there are 17, at least 17 clusters you identified.
Yeah.
How do you want to use those information in terms of deciphering pain or pain pathways, etc.?
Right. Two ways that we're going about this. One is by starting to work with Andrew Todd, who's kind of the world expert in dorsal horn neuron phenotypes and physiology and anatomy. So we're basically going through a lot of the antibodies that he's developed and worked with for decades now to identify different populations of dorsal horn neurons. And we're trying to use those antibodies to see if we can, place these neurons in space and understand what kind of afferent input they might receive. And then the other is using this kind of pharmacological interactome that we developed with the idea that, nociceptor subtypes or sensory neuron subtypes that express neuropeptides might likely communicate with neurons that express receptors for those neuropeptides. So we can see different populations that express different classes of receptors, and it'll take physiology experiments to do that, or to really nail that down eventually, but we're working towards being able to do those physiology experiments on human spinal cord. Great.
Thank you. Thanks again,
Thank
Dr.
you,
Ted
Stephanie.
Price, for joining us. Really appreciate it, and be well.
Yeah. Thanks, y'all. Bye.